And I am going to share my screen. Just need to find the right screen to share. Okay, here we go. So what we are going to do today, I'm just going to open chat. So if you have any questions, you can ask. My WebEx version has updated itself, so playing around with the new layout of it. So today we're going to go over the final exam logistics, and then we'll give a couple of announcements, and then we'll go through a final exam review, and we'll give some notes about the final exam review that we do today. So here we go. The logistics of the exam, and this will be the exam review, and hopefully you're not studying only 1 hour before the final exam. The exam takes place 1 week from today, so that is Tuesday, December 14th. The time will be from 2 until 3:50 p.m. Eastern Standard Time, and the value is 30% of your final grade. Now, the setup is I will be logged onto Blackboard, and then you log on by 1:50 p.m. because the exam starts at 2 o'clock p.m. Eastern Standard Time. Complete the online exam, and then when you're done, you log off and smile because you studied and did great. Of course, with academic integrity, it's our request that you adhere to the principles of academic integrity. So this means that you're performing the assessment yourself. You're not using other resources while taking the exam. You're not collaborating with other students while taking the exam, and you're not copying any material of the exam in any manner or fashion. And with the enforcement of that policy, I reserve the right to review the data analytics on each student's exam. And if there is a discrepancy that we note, I can investigate further, and you need to be aware that if we do have evidence that you have not behaved well and you have not adhered to principles of academic integrity, that you may have a charge of academic misconduct. If during the exam you experience a technical issue, you need to contact me immediately. And if you don't do so, you can't come to me after the fact and say, well, I had technical issues and that's why I wasn't there, whatever the case may be. You will receive a grade 0 for the evaluation. Now, the best way to contact me is via email, matt.kohler@georgiancollege.ca. So hopefully you have a secondary device also. So if you're using, would hope you're using a desktop or laptop computer, and for some reason internet goes down, then you have access to a mobile device with data or Wi-Fi on a mobile device that you can email me from that. If you call, chances are you're going to get my voicemail, but my voicemail goes to my email, so I'll be able to respond to you in order, because keep in mind, typically within the first 20 minutes is when we see issues experienced by students. So if you message me, you just may have to hang tight until I can get to your situation. Normally, the wait time is 2 minutes tops. The basic rules also are, if you are going to miss the exam, in order to be given accommodation, you need to contact me prior to the start of the exam and bring an official documentation for myself. So for example, if you're not feeling well, you need to go see a doctor or get diagnosed, and provide me with official documentation. And then the opportunity to write a makeup exam is up to my decision. It's up to my discretion. And the things that you're allowed are computer, keyboard, internet connection, and that big, juicy, smart brain of yours. So make sure that you have access to those things. So the topics that can be on the exam is basically what we have covered since the midterm exam. And so that would include the nervous system, acid-base balance, which we talked about, fluid homeostasis within the body, the endocrine system, hormones, cardiovascular system, which is right, the heart and the blood vessels, and also the respiratory system. The format will be multiple choice, short answer, fill in the blank, and diagrams. And each individual exam will consist of questions randomly drawn from a pool of questions. You will also have a longer answer question. So what that means is you will go through the first part of the exam, and then the last question, which is not worth any marks, would be, I understand when I when I can submit this portion of the exam, a Word document will open up in assignments and tests on Blackboard. You were to download that Word document, fill in your answer on that Word document for the question or questions, and then submit it to turn it in. And then that will be evaluated afterwards also. Study tips. Review the class notes and the textbook readings. So we have had recordings available throughout the semester. The recordings for the lectures would come up we had class on Tuesday, so they come up that afternoon after class, or on Wednesday at the latest, and would be available until 11:59 p.m. Eastern Standard Time that Friday. So you had access to the recordings for at least 3 days, but review the class notes and the textbook readings and use the textbook readings to supplement your notes. Learn the broader information first, and then learn the smaller details. Test yourself. A good way to do this is with the quizzes. So we still have some students who have not done all or even any of the quizzes. And those quizzes are worth a total of 10% of your final grades. That is there as a study aid for you. Practice with the quizzes. And then summarize your notes and do it again and again and again. In this course is the repeated exposure and application of the information, which will help you to understand that information. And don't wait until Monday, December 13th. Start now. And then if you have questions, you can email me. If we need to, we can set up an appointment and have an online meeting to discuss your questions or issues. Yeah, and the question from somebody was, it will contain long answer questions. Yeah, there is going to be a long answer question. So that will be, the instructions will be in the exam. That last question on the first part will be not worth any grades. When I submit this first section, a Word document will become available. I download the Word document, fill my answer in on it, and submit it to turn it in. Okay, so that's basically it for the review of the logistics. But we are going to go through some of the information that we've covered in this half of the semester, and we're not going to go through all of it, but we will go through a good amount of it. And basically, at this point, if you have any questions, feel free to put them in the chat or to unmute yourself and just let me know. Now, we had a question about, is it supposed to be done in the same time frame? Yeah, you have 1 hour and 50 minutes. In terms of how many questions, you have 80 multiple choice. And in terms of the ordering and the diagrams, I'd have to check. The long answer question will be worth anywhere from, approximately 20 marks. So the general rule is you will have approximately 1 mark per minute of the exam. So you have 110 minutes and you'll probably have about 100 marks on the exam. So of course, as a note, the Georgian College Fitness Center is open from Monday to Friday from 3 p.m. Somebody put they want 10 minutes more. Everybody gets the same time unless you have documented accommodation. And the weighting is 30% of your final exam or your final grade. So as a reminder, and I've told you this throughout the whole semester, and I'm talking to instructors who are teaching second semester students right now, they can tell students who are actively active, like are purposely active and exercising, and those who don't. And here's an example. An instructor has asked a student during a practical evaluation to place some weights on a barbell, and it takes a student 5 minutes to do that. This is a task that should not take more than 90 seconds. So it's obvious to the instructor that the student has not been regularly exercising because they're having difficulty with a simple task, which should be second nature by this point. So the Georgian College Fitness Center is now open. If you are in the region, you are welcome to use it. You need to be enrolled in courses. You need to present your one card to enter, and you must book a time to use the facility. In the province of Ontario, there are basically all fitness facilities are open. There are none that have not reopened unless they went out of business, unfortunately, which there are very few. And also, if you need help setting up a home program, you can contact myself or one of your instructors. But we really encourage you to be physically active and exercising in this program because we are fitness and health. Also the dining room. So again, if you're in the region, we do have the Georgian Dining Room and Bun on the Run. They are open this week and next week. So the Georgian Dining Room is open Monday through Friday, and it is open on Wednesday evenings. You need to show up, if you're coming for lunch, you need to show up right at 12 o'clock. And if you're coming for dinner, you need to show up right at 6 o'clock. And it is recommended you make reservations. What we find, especially with this week and next week, this is when they may start to get busy. So you want to make reservations. And students, you can eat a 3-course meal for, I believe it's $8. So you bring $10 and you get a 3-course meal, appetizer, entree, dessert, and you leave a $2 tip. That's cheaper than eating at fast food, say Subway. And also the food is all prepared fresh, and they do have vegan options on the menu. If you don't have, say, the hour and a half, you could go this Wednesday and Thursday or next Wednesday and Thursday to Bun on the Run. Both Georgian Dining Room and Bun on the Run are in the E building. Georgian Dining Room in room 100, Bun on the Run in 109. And Bun on the Run is food to go. Wednesday and Thursday, show up anytime between 11:30 and 1 p.m. and you pay for the meal, you order it, they give it to you, and you can go. So in terms of, let's talk about topics. These are the topics that we've covered in this half of the semester. And these are the associated chapters and the chapter page numbers. Now, we had in the beginning of the semester talked about active reading. You don't read your textbook as you would a novel for pleasure or a magazine. You read the textbook with the understanding that you need to understand and comprehend the information. You need to be able to recognize the words and what they mean. And you think about it, this is a technical manual about your body. So in a lot of ways, you're learning a new language. When we go into words such as homeostasis or alkalosis or osmolality, these are new words probably for a lot of you. So not only are you learning new information, you're learning a new language. It's the same with anatomy. When I taught anatomy, I would tell my students, you're learning the geography of your body in a foreign language because it's all Latin and Greek. And a number of the terms that we use in physiology have their foundations in Latin and Greek also. And so when you recognize that, you realize, yeah, it's a bit of a big task, but we've been exposing you to these terms for the past 13 weeks. And so hopefully these terms become more and more familiar with you. So we're going to basically go through some of the information. Now, the note here, what we're covering today does not represent all of the information you're responsible for on the final exam. You need to refer to the textbook readings also for that. And sorry, I just got a question about the long answer exam. So yeah, I can tell you right now. So we had a question about the long answer exam in terms of how do you answer it? There's on the midterm, I had two long answer questions. One was more of a process and one was comprehension. So I can guarantee you there's going to be one or two questions on the long answer question or segment of the final exam. For a comprehension question, you need to, it's worded in such a way that you shouldn't be copying off the internet. You need to give me your understanding of the concept that I'm asking you about. If it's a process question, there is more likelihood that you will be using a number of phrases similar to what's out there. But I always have at least one comprehension question where if you're copying from the internet, it's obvious. It's more of a, what is your understanding of this process? That's the style of question that I'll be asking. Okay. If you have specific questions about that, we can always set up a meeting. Okay, so again, what we're covering today is not everything that's going to be on the final exam. This is just more like an overview of the different topics. And today we may go a little bit rapid fire through the slides. So I won't be parking on one slide and explaining it in its full detail as I did when we first introduced the information. Having said that, if you have a question about a slide that we're on, feel free to put it into the chat or to unmute yourself and ask the question, and we can park there. With that understanding, because we already have gone through all of the information that we're presenting today, we might not get to the end. I've got 60 slides. That's a lot of slides. The only way I'll get through them is if I get to a slide and say, oh yeah, just keep in mind that you pay attention to this detail when you study this and move on. So maybe we'll get through it, maybe we won't. We might be here till 3:50 Eastern Standard Time. We might be done at 3 o'clock. I don't know. So this is how it goes. So let's talk about the nervous system. I mean, the nervous system, the function of the nervous system, and this is an image of, basically the body stripped away from the nervous system, is controlling and communicating. So the part of existing is you need to have communication, internal communication and external communication. right? You need to be able to receive information, communicate information from the external environment, but also from the internal environment. If you have a clogged artery in your heart, your body's going to give you signals. If you have indigestion, your body's going to give you signals. If you need to go use the restroom, the facilities, your body's going to give you signals. If there's danger outside, the environment will give you signals that the nervous system will communicate to the central nervous system so you can process that information. And the nervous system, it's quick and it's specific. So if I want to flex my elbow, right? So this is flexing my elbow, I'm bending my elbow, flexing my elbow. It's specific to the bicep brachii and the brachialis muscle. It's not really going to send messages to the quadriceps femoris. And basically what we do is we sense information, we process it, and then we have an output. And the output might be to the motor system or to the glands to produce a response. But really, there's two principal parts. You have the central nervous system, which is basically your brain and spinal cord, and the peripheral nervous system is everything else. And you'll either have spinal nerves that come off your spinal cord, or cranial nerves which come off of your brain. And there are 12 cranial nerves, but we don't test you specifically on that. The functions, right? The central nervous system is basically going to be controlling and integrating the information, and then the peripheral nervous system is where we start to get a number of sub-branches. So you're going to sense things, right, through special receptors or through somatic, visceral receptors, and that will go into the peripheral nervous system and into the central nervous system. But then you're also going to effect things, effect things, so motor division. And the motor division might be voluntary, somatic, it might be automatic. If it's automatic, it could be the sympathetic or parasympathetic nervous system. So sympathetic nervous system is normally mobilizing systems. It's getting you ramped up to do something. Parasympathetic nervous system is going to conserve energy and kind of keep things toned. And a good example of this is the heart. The heart has its own intrinsic rhythm of 90 to 100 beats per minute. The parasympathetic nervous system will slow that down. So average resting heart rate is 72 beats per minute. Some people, their heart rate at rest is 40 to 50. Or the sympathetic nervous system will ramp up the heart rate. So when the sympathetic nervous system activates the heart, it will bring its heart rate above 100 beats per minute, maybe as high as 200 beats per minute. You need to be aware of the different types of cells in the central nervous system. And be familiar first off, like we talked about, learn the broader information first and then learn the details. Well, learn what the names of the basic cells and neuroglia of the central nervous system. So these are support cells. And then once you've learned their names and you're familiar with their names, learn what they do and learn how they work. We also have the neuroglia, the support nervous system cells of the peripheral nervous system. So again, identify what they are and then be able to identify what they do. And we had shown you this, and this is a cartoon drawing on the left-hand side of a stereotypical neuron. And on the right-hand side, we see an electron microscope picture of a neuron. So you should be able to say, look at a blank image of a neuron and label it and tell us what the different parts are. Of course, we talked about the difference between a nucleus, a tract, a nerve, and a ganglion. So nuclei is a cluster of neuron cell bodies in the central nervous system, and the ganglia is a neuron cell bodies in the peripheral nervous system. A nerve are the axons outside the central nervous system, and a tract are the axons within the central nervous system. And then the different processes, right? The extensions. So we've kind of gone over this already, and how that works in terms of myelinization of neurons, and why do we have myelinization of neurons? And you should know the basic types of neurons and the comparison of them. In terms of neurons, we say that neurons are excitable. And what does that mean? Well, the inside is going to have a different charge than the outside due to these ions. And ions are basically elements or electrolytes which are missing an electron or have an extra electron, right? So in this case, we talk primarily about sodium, potassium. So if we allow these ions to go in or out of the cell, the voltage difference can change. And then that's why we talk about excitable, right? There's a change, it's excitable. So you should be basically familiar with what's going on. You have more sodium channel ions outside of the neuron than inside. You have more potassium ions inside compared to outside. And we're going to have these different types of gates. You have chemically gated gates, which there's a difference in the voltage of the cell. It'll open these gates, or sorry, chemically gated gates. Sorry, if it receives a chemical signal, it will open up. The voltage gated gates or channels, if there's a change in the polarity or charge of the cell, it will open up these gates. We also have mechanically gated gates, which will open and close in response to physical deformation. And so be familiar with what depolarization is. Depolarization is when we see the inside of the cell go less negative or goes towards the positive side. So we can see in the middle, we see that this has gone up a little bit. They're going a little bit more positive, and so that is technically depolarization. If it's going a little bit more negative, that's hyperpolarization. And we had talked about graded potentials and action potentials. So graded potentials happen in the dendrites and the cell body of a neuron, and they kind of diminish over distance. So they get weaker over distance, so it doesn't travel that far. But an action potential travels quite far, and they're quite resilient. And of course, a neuron is either going to send a signal or it's not. There's no in between. But what determines the difference in, say, pain sensation is the frequency of those signals. The synapse is the connection or the junction between from one neuron to another, from a neuron to a gland or a muscle cell. And of course, we have the axosomatic synapses, we have axodendric synapses, and we have axonal synapses. So axosomatic is when an axon goes on the cell body. Axodendritic is when an axon goes onto the dendrites, and an axoaxonal is when it goes from an axon to an axon. And it could be chemical or it could be electrical. I mean, electrical we find more, they're less common, but they're more in some specialized regions of the brain and more abundant in embryonic tissue, but chemical are more common, right? And this is what we also learned about with skeletal muscles prior to the midterm. We also talked about that you can have an excitatory postsynaptic potential or an inhibitory postsynaptic potential, where basically with an excitatory postsynaptic potential, we're priming the neuron to be easier to have an action potential. And with inhibitory postsynaptic potential, we're inhibiting, like we're making it harder for an action potential to go. And again, you may have two excitatory postsynaptic potentials, which will not lead to an action potential, or if they're close enough, they will lead to an action potential, or they could summate, like they are one on top of each other, and it leads to an action potential, or an inhibitory postsynaptic potential will prevent that action potential from going. Okay, so that's rapid fire through the nervous system of the body, but the whole idea is that you're going to get signals from the nervous system. The whole idea with the nervous system is you're going to have basic communication. So you see something, you see a car coming down the road, and you can see it and you can hear it, and that sends signals to the central nervous system, and then that tells your muscles to stay, to just perform isometric contraction. So you stay at the side of the road until the car passes by you. And then you see it's clear, and now your nervous system is sending signals to the muscles so you can proceed to walk across a road. So it's communication. And the whole idea is that these cells will be more negative on the inside than the outside. So by sending those signals, they change. It's like a ripple wave
of depolarization that travels as they are receiving signs from the external environment. Okay. So we're going to go on. So the difference is, though, with hormones, hormones are longer lasting. They take usually longer to take effect, and they're more general. So whereas with the nervous system, we talked about the nervous system sends a signal to the bicep brachii and the brachialis to contract, and they contract, and you flex your elbow. With the endocrine system, you could release hormones, but it doesn't just affect, say, the bicep brachii. Any cells which have receptors for those hormones will be affected by the bicep brachii or by the hormones being released. So you need to be familiar, again, when we talk about learning the broader information first, learn basically the names of the glands and their locations, and then learn what hormones they release, and then learn what stimulates those hormones to be released and what effects those hormones have on the body. In terms of the receptors we talked about, we have amino acid base hormones that are basically water soluble. Well, yeah, and so these ones cannot get through the cell membrane, but they will start a cascade. And you should be familiar with the order of events in this cascade and in this cascade. So the cyclic AMP and the PIP2 calcium signaling mechanism. So this is for amino acid-based hormones. And then you have steroid-based hormones. And these actually can go through the plasma membrane and then bind to a receptor within the cell. So you should be familiar with the steps here. Stimulation, it could be stimulated by a difference in blood levels. So in this example, we have low calcium levels in the blood that activates parathyroid glands, which releases PTH, which will activate a release of calcium from stores in the body. It could be neural stimuli, right, from the central nervous system. And it could be hormones acting on other hormones. So we have hormonal stimuli also. And a hormone will only affect a cell which has its receptors for it. And we could see things such as upregulation or downregulation. Now, a lot of the endocrine chapter we spent saying this is the hormone, here's what it releases, here's what it does. And really to study for that, you just need to review it and test each other on it. So if you have formed, I understand, we always see students form WhatsApp groups, then test each other. Try to teach each other and test each other on it. When you can teach something, then that demonstrates that you have a good understanding of it. Also, be careful because this is the time of semester where I'll get comments from students who will say, oh, well, I talked to one of my seniors. Like I talked to someone who's in semester 2, 3, or 4 of the program, and they told me this. Sometimes your other students get it right, sometimes they get it wrong. So you want to make sure you have the right answer. So if you have a question, feel free to email me and I will do my best to answer. Acid-base balance is basically about fluids within the body. So, we talked about you have maybe on average 40 liters of fluids within your body, and 25 liters of that is in intracellular fluid. The fluid outside of the cells, that's 40% of your body weight. The interstitial fluid is the fluid immediately outside of the cells, or sorry, intracellular fluid, is fluid in the cells. Interstitial fluid is fluid immediately outside of the cells, and then you have about 3 liters in the plasma. So you should be familiar with those different categories and be familiar with what is where. When I first introduced this course about 13 weeks ago, I said, try not to learn things in isolation. Well, if you think about excitability of the neural cells, well, why are they excitable? Well, you have more sodium ions outside and more potassium ions inside. And then you look at this chart and you go, it makes sense. So if we were to look down here, we see with sodium, well, we have a lot more sodium outside of the cells than inside of the cells. And with potassium, we have a lot more potassium inside the cells than outside the cells. So we start to connect these thoughts and we go, okay, this makes sense. There is going to be set pathways, if you will, for certain elements and materials within the body. Oxygen goes from the lungs to the blood, to the interstitial fluid, to the cells. Carbon dioxide goes from the cells, the interstitial fluid, to the blood plasma, to the lungs, and you exhale it. Gastrointestinal tract, nutrients go from the gastrointestinal tract into the cells. Water can travel both ways. And with the kidneys, water can travel both ways or go into the kidneys to get excreted from the body. And the same with nitrogenous waste. And this nitrogenous waste are typically formed when your body breaks down proteins for whatever purpose. On average, you're going to have an intake and output of 2,500 milliliters or 2.5 liters of water, or fluids, I should say. The intake, 10% of that's from metabolism. If you think back to week 3 with the electron transport chain, you produce water as part of your metabolism. Foods, you get about 30%, and then beverages, you get about 60%. And then to get rid of that urine, you're going to be excreting about 60% of your fluids through urine, which will carry metabolic waste products. You lose about 28% of it through insensible loss, right? You just don't feel it leaving your skin and your lungs. 8% of it you sweat out, and about 4% of it you poop out. And we talked about disorders of water balance. So you need to be familiar with osmolality, right? We have dehydration, you have hypotonic hydration. We also talked about edema. So edema is when you have a unusual accumulation of interstitial fluid where you get swelling around the cells. So it's not the cells actually swell, but it's around the cells that swell. And then try to understand the basic concepts between or regarding chemical buffer systems. Our body tries to maintain a pH, depending on the system, of about 7.35 to 7.4. So a pH of 7 is neutral. If you go to 14, it's extremely basic. If you go to 1, it's extremely acidic. And our body tries to maintain just slightly basic. So 7.35 to 7.45, depending on whether it's the interstitial fluid or the blood. We talked about the dissociation of different acids and bases in fluids. And we got to this chart, and this chart was like, oh, that's complicated. What's going on here? Well, here's my recommendation. Study this type of chart in conjunction with the respiratory physiology. Because if you recall, and this is now where we tie them in, we actually go a little bit more in depth as opposed to the rest of the stuff we skimmed over. If you recall, to transport carbon dioxide, a good majority of it is dissolved in the plasma, but to dissolve in the plasma, the carbon dioxide has to dissociate to a certain degree. So carbon dioxide is associated with pH balance within the body. And this is a good thing because your body will respond to the hydrogen ions within the blood. So if we sense an increase in hydrogen ions with the blood, we'll increase ventilation. And if it's long-term, it may increase excretion of certain elements through the kidneys to help maintain that pH balance. And we can say the same thing for here. Okay, cardiovascular physiology. So I'd said, this is kind of my jam. I'd done grad research with heart transplant patients, heart failure patients. And I mean, in simple terms, the heart's a pump, but it does more than that, right? So again, if you want to learn the parts of the heart, label the parts of the heart as blood flows through it. And you have your systemic circulation and you have your pulmonary circulation. Systemic circulation, blood goes through the body. Pulmonary circulation, blood goes past the lungs. And of course, you also have coronary circulation, which is the circulation of blood around the heart. You should know, even though we covered skeletal and smooth muscles prior to the midterm, you should know the differences between cardiac tissue and skeletal and smooth muscle tissue. You should be familiar with the order of events in the conduction system of the heart, and also why and how it's modified. Also, differences in the excitability of cardiac tissue as opposed to skeletal or smooth tissue. And for a normal ECG, I should be able to put this up blank and say, what does this, like have a circle around one part and say, what does this part represent? Or what does this part represent? And you should know what part of the ECG or EKG represents atrial depolarization, ventricular depolarization, atrial repolarization, ventricular repolarization. And when I say that, so this is a good example. I say atrial depolarization, like, oh, wow, atrial depolarization, they know what that means. Atria, the two smaller, higher chambers of the heart, which receive blood, either from the lungs or from the body. Depolarization, when they change in their charge, right? When an action potential goes through them. So atrial depolarization is when the charge of the atria changes, but you see depolarization before you see contraction. They depolarize and then they contract. This is a bit of a sock drawer, but basically we're looking at here in the middle, we're looking at the pressures within the aorta. That's the biggest vessel that leaves the heart. The left ventricle, the chamber that pumps blood to the body through the aorta, and the left atrium. That's the chamber that receives blood from the lungs, passes on to the left ventricle. So we're looking at pressure changes in the middle and volume changes in the left ventricle at the bottom. So you should be familiar with this concept also, and with the concept of cardiac output. Cardiac output is how much blood the heart, really the left ventricle, pumps out per minute. And that is determined by heart rate, the number of contractions per minute, and stroke volume, the amount of blood that is pumped out per contraction. And then respiratory physiology. So respiratory physiology is really just talking about, delivering and getting rid of gases. So we're taking in oxygen, getting rid of carbon dioxide, and the various constituents for it. So you have the various parts of the trachea and looking at the ciliated cells, right, help clean out the system. Looking at the alveoli, these are the air sacs where gas exchange occurs, and they are working in conjunction with the capillaries. So the alveoli are basically where air can be exchanged, oxygen can go into the blood, and carbon dioxide can come out of the blood. You should be familiar with the basic concepts or the different types of breaths that you take, right? So tidal volume, what you're doing right now, normal, inspiratory reserve volume, how much air can you take in your lungs, expiratory reserve volume, how much air you can blow out of your lungs, the residual volume was always there, and then the combination of those, right? The inspiratory capacity, the vital capacity, total lung volume, functional residual capacity. The basic properties of gas. So here's the idea. Even though you can't sense it, we are being subject to about 1 atmosphere of air pressure right now. So that's approximately 760 millimeters of mercury. If you are at sea level, it should be about 760 millimeters of mercury. If you're in the Himalayan mountains, maybe at 5,000 meters altitude, then that will be less than 760 millimeters of mercury. But that 760 millimeters of mercury is made up of the different gases in the air. Nitrogen being the big one, 78.6% of the air is nitrogen, so that makes up 78.6% of the pressure. Oxygen is about 20.9%, that makes up 20.9% of the pressure. And the idea, the concept had been, well, if we take 2 cylinders of gas, one has 2 atmospheres of pink gas, the other has 4 atmospheres of purple gas. We put those both into a same size of cylinder, we now have 6 atmospheres of gas. This diagram is basically showing that in this flow chart here, the x-axis is the time that a blood cell is in a capillary in the lung, and the y-axis is basically the pressure of oxygen in that blood. So in this case, when the blood cell enters the pulmonary capillary, within 1/4 of a second, it is fully saturated with oxygen. On the right-hand side, we see the differences in oxygen, carbon dioxide in the blood, depending on where it is. In the alveoli of the lungs, there's 104 millimeters of mercury of pressure of oxygen, 40 mils of mercury pressure of oxygen, carbon dioxide. In the pulmonary veins, it's about 100 to 40. At the tissue, it's less than 40, greater than 45. In the blood leaving tissues entering the lungs, it's 40 and 45. And inspired air is 160 and 0.3. You should be able to explain this diagram here. So what we have going on here is here we have the pressure in millimeters of mercury of oxygen. And here we have the percent saturation. So when you have 100 millimeters of mercury pressure of oxygen, you're going to have about 100% saturation of the red blood cells. Like you're carrying as much oxygen as your blood can handle. If you drop down to 40, we see that it drops down to 80%. So it's not linear, that we can have a 60% decrease in the pressure of oxygen and only a 20% decrease in saturation. But if we go below 40, if we go 40 to 20, now we find that we have about a 40% drop. We've gone from 80 to 40% saturation. So the idea being, this is the oxygen hemoglobin dissociation curve. So the idea being, you can have a big drop in pressure and not a large drop in oxygen saturation. But once you get to 40 millimeters of mercury, once you start to decrease, you start to see a big drop in saturation of the blood. And again, so refer back to the acid-base lecture. The role of carbon dioxide is linked to the balance of pH within the blood, because we see that when carbon dioxide goes from the tissues into the interstitial fluid into the blood, well, some of it's dissolved in plasma, but some of it will dissociate into hydrogen ions. And some of it down here will dissociate into hydrogen ions. And if you have a lot of carbon dioxide, you're going to start breathing faster and deeper to try and blow off that, because you have a change in pH. More carbon dioxide in the blood means more hydrogen ions, which means a lower pH, which means you're becoming more acidic. And so that's basically it. I mean, that was quick, that was rapid fire in terms of our presentation of the information for the semester. But again, we haven't presented everything. We've given you more of an overview of what's involved in this semester. So for this week, there's no lab. There's no lab next week. We have the exam next week, Tuesday, December 14th at 2 p.m. Eastern Standard Time. So what I would encourage you to do is to review your notes. I understand you have other assignments and exams. That's the nature of this college season. However, having said that, if there are questions, you can email me your questions and we can set up an appointment if we need to, so we can go more in depth with the information. I would also encourage you